introduce the chief guest. Sir, it is up over to you, sir.
Hello everyone, welcome to today's session. It is a glorious moment to extend my warm welcome on behalf of the Department of Biotechnology, and I want to convey my heartfelt gratitude to the Velmurugan College for mission and over the seminar on biotechnology. 2 days scheduled versus upon a spectrum of exciting and benefiting themes that discuss the aspects of biotechnology in human life.
I would like to introduce today's presenter, Dr. Velmurugan, who is working as a scientist at KMCH Research Foundation at Kovai Medical Center Hospitals, Coimbatore. So, coming to this Velmurugan's study area, he did BSc Plant Biology and Biotechnology at St. Joseph College, Tiruchirappalli, and later on he moved to MKU. He did MSc in Biochemical Technology, then he got awarded a PhD in 2015. So to his credit, he got many awards, especially I would like to tell you very few, because in his CV he has given a lot of things. So he got SERB Startup Research Fund Grant Award from DST in 2020. Then Young Scientist Award in 2017 from the Association of Microbiologists of India. Then he got Institute of Postdoctoral Fellowship from Indian Institute of Technology, Chennai. Then he was awarded International Max Planck Research Fellowship in 2008 from Max Planck Society, Germany. Then he was the first time holder and won Merit Endowment Awards during his undergraduate studies. Coming to the research areas, so far he has published 14 manuscripts. Most of them are well credited and more than 8 impact factors he got. Then he has published 2 book chapters in 2 different books. One is from the Elser publications and another one is Springer Verlag publications. Then apart from that, recently he is having enough fund from his credit. So coming to this ongoing and completed projects, as a principal investigator, he recently got 2022-23. He received an Interference Center for Promotion of Advanced Research Center DST, 1 crore and 90 lakhs thousands from the Indo-French Center for Promotion of Advanced Research. Then he got 22 lakhs from the SERB DST fund for the year 2020-22. These are all ongoing projects. Tumor microbiota in present breast cancer, implications on pharmacogenomics of anti-cancer drugs. Then in the same, he got from the same Institute of KMCH Research Foundation, he got 9 lakhs for the investigation of the effect of microbiota and pesticides on the efficacy of oral hypoglycemic drugs. Then 2017-19, he received another 6 lakhs from the KMCH Research Foundations for the rural epidemiology of diabetes in South India studies. Apart from that, he also served as a co-principal investigator from ICMR, ESTMs and KMCH Research Foundations. So like that his CV is going on. So today's his talk is, the title of the talk is, he is going to talk microbes, a key driver of the world. Okay. So everybody knows that generally microbes has a broad spectrum of 7 types. Mostly it includes bacteria, archaea, protozoa, algae, fungi, viruses, and multicellular animal parasites. So each type has a characteristic cellular composition, morphology, locomotion, and reproductions. So most of the microorganisms are beneficial in producing oxygen, decomposing organic materials, then providing nutrients for plants and maintaining human health. But at the same time, some can be pathogenic and causes disease in plants and humans. Okay. So these microbes play a key role in nutrient cycles, biodegradation, then biodeterioration, climate change, food spoilage, the cause and control of diseases, and biotechnologies. For example, if you take a human body, it hosts 10 microorganisms for every human cells. And these microorganisms contribute to digestion, it produces vitamin K, it promotes development of the immune system and detoxify harmful chemicals. And of course, microbes are essential to making many foods with the enzyme we enjoy, such as bread, cheese, wine, like that. So now I welcome the Dr. Velmurugan for his speech.
Okay, thank you, sir. Thank you for the excellent introduction. Hope you are all able to hear me. So, maybe without taking much time, I will start my presentation. So are you able to see my screen?
Yes, Velmurugan.
Yes, sir.
Okay. Thank you. Thank you all the organizers, and it's been a proud moment. So today, as Paniswaram sir has described, today I will be presenting on microbes, the key drivers of the world. Okay. So before entering into the talk, I would like to thank all the organizers. It's been a proud moment to present in a prestigious century-old college. So though I am not directly associated, linked with the college, but in several ways I have been associated with most of the faculties, including the coordinator. And yet another thing I want to share here is my first PhD student is from your college. He did his BSc Biotechnology from your department. So that's yet another thing I want to share with you all. So going into the talk, I planned the talk in a basic level, so describing from who are microbes, where are they, what are they doing, and then we will slowly move into some more detailed parts. So when you ask who are microbes, we can describe them as for all microscopic. So it starts from viruses, bacteria, and then both spans into animal and plant kingdom, protozoas, fungi, and microalgae. So all these who are in microscopic size are the microbes. So their size ranges from, if you see the proteins, viruses, their size ranges from around 50 nanometer and up to bacteria up to 10 microns. So these are the microbe size range, and so they are diverse in size, shapes, as we have seen their size. So they are spherical, so they have rods and different dimensions, and you can see the various viruses which are of different size and shapes are present. And where are they? So they are omnipresent. So the microbes are present everywhere. Okay? So they are present in everywhere, even the places where the humans cannot survey or any other living beings cannot survey. Even there are reports that microbes are present even in the extraterrestrial spaces in Mars or Moon. There have been several explorations, and they are present everywhere. Okay, and if you see, the major habitats are nearly, you can see more than 1/4 of the microbes are present in the soil, and the rest are largely they are present in deep oceanic surfaces and below the earth. So this has been a estimation, and the role of these microbes are largely unexplored. What I'm going to speak on the microbes in our human soil or water or humans or constitutes a very small proportion. Okay, so these are all constituting a very small proportion, but the major habitats are residing deep in the ocean and in the earth, whose function is really unexplored. So as I said, today, as these function of these microbes are largely unexplored, we will focus on the microbes which are associated with the different parts of biosphere. To take the plant kingdom, starting from algae to angiosperm, they known to be associated with all the plant forms. So they are present in the soil, they are present in the roots, they are present in the leaf surface, and they do everything. Okay, so they help. And similarly, in case of animal kingdom, also they are present in all the different classes, different strata of the animal kingdom, starting from protozoans, porifera, annelids, echinoderms, and mammals. Everywhere it is present in the animal. So a very good example is sponges, which comes under the second strata of the animal kingdom in the porifera. And the sponges are described as the microbial fermenters. They are immobile animals. They will just look like a plants inside the deep oceans, and nearly 90% of the body weight is constituted by the microbes. So they are, as I said, they are sessile, they are immobile, they cannot move. Just like a plant, it would be rooted in the surface. So the water will enter through this pores, and then it will release through the pores, so through which it will absorb the nutrients. And they have a lot of microbes which are residing inside their body. And once sponges were considered to be just like medicinal plants for different chemical products, compounds with medicinal activities has been isolated from sponges. Now they have been proven they are not really from the sponges, they are produced from the bacteria residing inside the sponges. Okay, so it's a very good example to show how it was associated. And yet another thing is the insects have a wide, it's one of the major animal which have large number of microbes residing in all throughout the body, from its salivary glands, midgut, and everywhere. And even there are studies which shows that and even the ICMR Institute is Madurai is involved in identifying the microbes in the gut of bacteria as a measure to combat dengue infections. So coming to human system, okay, so this is a very strong statement that human biology can no longer concern itself only with human cells. So this is what the physiology, either as the biologists or the medical MBBS graduates who study the organs and the different physiology of the human system. So, but you can see the microbes are present throughout the body, and this is what makes the complete human. But largely, we are studying only about this, and we understand the function of these only. So you can see what is the importance of these microbes through this diagram. So there are only 30 trillion human cells, but at the same time, our body harbors more than 100 trillion microbial cells. So, and in case of the genes, we largely speak about the genetics and the human genome. Only a very minute proportion, maybe a less than 1% is the human genes which are present in our body. There are more than 99% of microbial genes, the same is here represented. Microbial genes which are present in the human body, whose function is largely unrecognized, or we are not aware of that when we are considering the human diseases or the day-to-day activity. So you can see in our body weight, maybe the human cells are larger in size. So in case of physical weight, it may be just 3 to 4% of the weight is contributed by the microbiota. But when you come to number of cells or the number of genes, you can see the microbiota outnumbers the human system. So has been a lot. So the other question is, can we isolate and grow all? So probably you students should have your practicals should have did with the isolated E. coli and grown E. coli at 37 degrees. So practically speaking, we don't know to grow all the bacteria. The bacteria that we have known to grow is a very tiny portion which we know to grow in LB or some nutrient media, and then we do some cultures and then we grow it at 37 degree, which is a physiological temperature. But there have been bacteria which grows at even at extreme high temperatures like Thermus aquaticus, from which we got Taq polymerase, right? So like that, there are, so how we know now that there are several bacteria are present. So now we have uncultured genomics, so in which we just, as we doesn't have the practical knowledge to isolate the bacteria, we will just destroy all the cells and we will isolate the DNA. Okay? So from the DNA we will identify what are the bacteria which are present, through which is now famously called as metagenomics. So we have identified different groups of bacteria present in different environments, from water, soil, and in the human body. Okay. So what they do? So we have seen the microbes are present everywhere, starting from soil, from the abiotic surface to plants, animals, and the human body. And what they are doing, right? Are they just simply presenting and exploiting us, or they are really doing something? So the thing is, so especially during this COVID-19 pandemic, you can see the advertisements everywhere. They are mentioning for all the products. Even I have seen some advertisements for wall paints, and everywhere they are mentioning it kills 99.9% of the microbes. So, but in reality, not all the microbes are not bad. Okay, so I will say 99.9% are good. Maybe less than a 0.1% are only disease causing. Okay, that's true due to human errors. They are not really, for example, this COVID virus or this has been prevalent in this world for centuries, but now has becomes a pandemic due to human interventions only. So they are the key drivers of the world. They they produce a lot of carbon dioxide. Nearly most of the photosynthesis done by the is largely from the microalgae which are present in the deep oceans. Okay, so they produce fossil fuels, they produced rocks, they created soils, and they do everything. Okay, they are present in the atmosphere, they are present in the clouds, they play a role in the rains everywhere. Okay, so coming to the plant system, so they do a lot of, they are the real biofertilizers. In the term of chemical fertilizer, now we have destroyed the soil microbiota, but they play an important role in for the microbes to take up. They convert insoluble phosphate to soluble phosphate form, and they play a role in ion uptake, and they play a lot of role in overcoming stress conditions and helping in providing nutrients to the plant. Coming to the humans, so it's not that only during a disease condition microbes are present. It is always present with us. It is born with us, and even after our death, it plays a role in decomposition of our dead bodies. Okay, so it is always there present. So a balance between the microbes, when we have a deterioration of your microbiota due to prolonged intake of antibiotics or due to other conditions, that determines the human health. Okay, now the last 1 decade there has been different studies which shows that the type of food we are intaking, the environment we are getting exposed, are making a changes in our microbiota. So they are present everywhere. Not only the bacteria, there are phages which determines the phages are the viruses which kills the bacteria. So the number of phages which are present, different types of phages determines the human microbiota. So it is present everywhere. Not only in human health, they plays a major role in determining the action of chemicals. When I say chemicals, it varies from different things, from the simple medicines we are taking to environmental pollutants. So we can see that not all the medicines work for all the people, right? So some people have good response to medicines, while some people doesn't have response, and the same time sometimes for some medicine some side effects are reported for some people. So these microbiota plays a role. They make some drugs inactive drug into active one, and then it makes, sometimes it converts into a toxic metabolite, and like that there has been different things has been entering into the, just in the last few years it has been understood how the thing, as I said. So this is my work with I have did for my PhD in Madurai Kamaraj University. So I worked on pesticides, okay, pesticides and diabetes, and surprisingly we found when the pesticide was metabolized by the microbiota, so it's a group of, I think last webinar you had a talk by Dr. Sundaravadi, who we presented in a similar topic. She has been associated with me. So I was working on how these pesticides are causing diabetes, and we have proved that these group of pesticides called organophosphates, right? Okay, and these are the largely used pesticides today. Nearly more than 60% of pesticides which are available today in market belongs to this group of organophosphates. Okay, while endosulfan DTT comes under organochlorine pesticides, which have a long half-life time, years of half-life time, and persistent one. Whereas these organophosphates have a short lifetime and they are biodegradable. As a result, they become a largely used pesticides in the last few decades. So you can see these pesticides, as I said, they are biodegradable through this star represents the pesticides. They enters our gut, our system through the food as they are contaminated and heavily sprayed with different pesticides, and these bacteria converts them into some gluconeogenic substrate like acetic acid, which is then converted to glucose. So this was a complete study has been done from starting from an epidemiological level by understanding how much diabetes is prevalent among the farmers who are exposed to pesticides, and then followed by animal studies, and then at the gene and the molecular level we have studied, and we have proved that the gut microbiota plays a role in this pesticide-induced diabetes. So this was published in Genome Biology, which has an impact factor of 18.5. So in our study, we have showed that pesticides are metabolized by the microbiota. Similarly, different groups from different parts of the world have showed how microbiota, microbes residing within us, plays a role in metabolization of the chemicals like arsenic or different organic pollutant. Bisphenol A is a compound which is present in plastic. So you can see the plastic bottles, like for children feeding bottles, it would be mentioned as BPA free. Okay, BPA is bisphenol A, which is a endocrine disrupting chemical and a very toxic compound. Okay, so maybe the plastic bottles was we are using are largely having this BPA. That's why you can see specifically the feeding bottles mentioned as BPA free. And saccharin is artificial sweetener, okay, which has been proposed as an alternative to sugar. So you can see in advertisements and everywhere for diabetes people, even many medical doctors are suggesting, which is present even in Coke and Pepsi. Okay, but there has been shown that these saccharin are inducing diabetes through microbiota. Okay, and these emulsifiers are present in cakes and other substances. And we have also did another study on another compound called 4-methylimidazole, okay, which is a coloring agent present in Coke and dark chocolates for that coloring. So we have showed that it induces pancreatic cancer, which are not presenting today, which also work done in Madurai Kamaraj University. So based on all this, we have proposed that at present, when a drug or a chemical is induced, it undergoes a toxicological risk assessment to make sure that it doesn't harm humans. It's applicable for even vaccines also. But though has been a lot of knowledge has been generated on microbiota, but there has been no there is no inclusion of microbiota in the toxicological risk assessment. So in our recent paper, we have proposed that microbiota should be included in the toxicological risk assessment to understand what changes caused by the drug on the microbiota, and at the same time how the chemical is getting modified, because we have seen in case of this organophosphate pesticide that pesticide is being converted to a gluconeogenic substrate that is being converted to leading to diabetes. So this is a brief workflow we have proposed. So coming into the current scenario, so COVID-19 pandemic and microbiota. So now we are all just sitting in front of the screens, which we never thought before exactly 1 year before, because in the last January only, COVID-19, it was detected in December 2019, so it has been termed as coronavirus disease 2019. That's why it is shortly called as COVID-19. So 1 year, exactly 1 year back, we never thought it could create this much situation which never we have heard or seen in our lifetime. So how this microbiota associated with this COVID-19 pandemic? That's what today I'm going to discuss. So I'm going to speak on two different aspects. So what is the role of microbiota which are present in our body plays a role on this SARS. So the virus is called as SARS-CoV-2. Okay, the disease is called as COVID-19. The causing virus is called as SARS-CoV-2. So and then the other aspect is, so in case of just protecting ourselves from this COVID-19 infection, there's been a lot of disinfectants has been used in the last 1 year. So right from whether it is from the starting from the human skin, and it has been sprayed everywhere. I have seen it was sprayed on when I have seen the Chief Secretariat building at Chennai is being sprayed completely with sodium hypochlorite. So it has been released everywhere, right from buildings, water bodies, soil, everywhere. It has been lot of things has been sprayed everywhere, and we are also using till date. We are using a lot of things on our using these disinfectants. So today, first we will see about which is the work done by us to understand the role of microbiota. So when I said the microbiota is present everywhere in our body, the stomach and the intestine is a major reservoir of the microbiota. Okay, and a lot of the studies, including our study on the pesticides, has been largely focused on the gut microbiota. So besides the gut, now there are reports microbiota is present even in the respiratory tract, and even we are now working on a project where we are trying to explore the microbiota present in tumor tissues, in the breast tissues. Okay, so microbiota in case of this upper respiratory tract, it is present there. So as you know, all of you have understood, this SARS-CoV-19 virus is being detected in the nasal swabs, right? In the not from the blood, or in the blood only you can detect the antibodies. So they are present, and this upper respiratory tract is a natural reservoir of the microbes in our human body, and there is a lot of studies which show that these microbes play a key role in the gut, and they send signals, and they plays a key role in pulmonary health and diseases. So we thought, and in the parallelly we have seen not everybody got infected with this COVID-19. Person who is staying in the same house, the husband is infected, but the wife is not infected, and not all the infected people had serious complications. Not all the people who had serious complications need medical interventions, and not all the people who had a serious complication entered mortality. So that we have seen a huge variation in the COVID-19 outcomes. So we thought maybe the respiratory tract would be playing, the microbes present in the respiratory tract would be playing a big role. And it has been well known, these commensal, there are a lot of bacteria which are present, which are largely termed as commensal. So you have known symbiosum, so symbiotic bacteria, but lot of which gives and takes something from us, which is gets food nutrition from us, at the same time it do some good thing to us. So commensals are generally termed as just they gets the nutrition and they doesn't do any harms to us, but in general I would say most of them are only symbiotic. Okay, so and we know that these nasal microbiota also gets affected due to antibiotics or during viral infection. And recent study has showed in 2019, it was published, they have showed how influenza virus and vaccination causes the changes in the microbiota. And there has been several commensal bacteria which are present, better to term as symbiotic bacteria present in our nasal tract, Corynebacterium, which produces some antibacterial fatty acids. They themselves are bacteria, and they produce some free fatty acid which inhibits pathogens like Streptococcus pneumonia. And there has been a recent study which has been ecological study where they have identified a similar group of frogs present in a wild community, okay, in forest. Frogs which are present in one region got affected with a viral infection, while the frogs which are present in another region was not affected. When they studied the two groups of the frogs, they understood that a group of microbes which are present in the skin of the group of frogs produced resistance to this viral infection. So based on this background, we questioned ourselves why the nasal bacteria could provide the resistance to the people, or could play a role in the different outcomes due to COVID-19. So, and also it has been well known that most of the deaths which happens during this COVID-19 are due to viral infection. We can have seen like VIPs like SPB or have died, but it has been declared the people can visit his body since there is no COVID-19 infection was over, but he has died due to secondary bacterial infections. So there has been a lot of studies which showed that during influenza, the micro secondary bacterial infection, and now only there is a little bit data on the role of secondary bacterial infection during COVID-19 mortality. Whether due to this COVID-19 mortality, these pathogenic bacteria has becoming more potent and they are causing the disease. So as I said, these are the different questions we asked ourselves. So does microbial diversity of the upper respiratory tract of a person determine his susceptibility, recovery, or response to treatment or mortality due to SARS-CoV-2? Does this virus infection disturbs the microbiota of upper respiratory tract? Does this disturbed microbiota leads to proliferation of pathogenic bacteria that adds to morbidity and mortality due to COVID-19? So if we can understand all this, can we develop specific antibiotics which can target a single targeted species, or is it possible to recover the lost symbiotic bacteria just like probiotics? That's what our question. This is how we have started this study, and since this is a very potent virus, there has been a lot of restrictions, and we need called as BSL 4 facility, biosafety level 4 facility to handle this virus. So since in our institute even BSL 4 is present just in 3 institutes in all over India, and now only Tamil Nadu Veterinary University is establishing the facility. So we came across this publication from a US group who has worked on using this. So generally lab animals like mice or rats are not susceptible to this COVID-2 SARS-CoV-2 virus. So they use this animal model called ferret, which is taxonomically closely related to cats. So they have used this, and they have published an article on how it makes changes in the host gene expression. Okay, they have collected nasal washes after infection, and they have studied in the nasal epithelial cell what genes has been expressed. So we established a collaboration, and whenever people study on the host transcriptomics, always there will be a set of when I say transcriptomics, it will be millions of reads of RNA will be present. So they will just match with the human genome. Always there will be a 3 to 5% of unmatched reads. People generally don't care about that. They will just discard it. So we just took up those reads, and so this is the what they have did. So they have included a influenza viral infection also, and they have collected nasal wash at different timelines. Okay, and then they sequence. So they have deposited in a public database in PubMed. So we have downloaded a sequence from that, and then we mapped the unmatched reads which are not matched with the ferret genome to the microbial genomes. And what we have found is this is similar to what they have reported in the case of host transcriptomics. They have reported influenza infection. This IVIV is influenza. Okay, they have reported that during influenza there has been a major change in the host transcriptomics, but in case of SARS-CoV, there is a less or very few changes has been reported. Similarly, you can see. So these are the different group of bacteria. Just like when when we read in in taxonomy like protozoa or porifera. These are the different bacterial phyla which are present, proteobacteria, bacteroids, firmicutes, which are at the phyla level. So you can see this light blue gamma proteobacteria has been proliferated during influenza infection, but you can see it is not, still it is increased, but there is not a major change. But you can see still there are many differences at the within, so you can see chlamydia has got reduced. So like that at the phyla level, there has been differences. So next what we did is we just studied at on day 7, just at the orders, different bacterial orders, how it has been changed. You can see there has been this Burgdorferi. So we see an inverse correlation between influenza and SARS-CoV-2. Both are a viral infection which are attacking the lungs, but we see the microbiota shows a differential thing. You can see this Burgdorferi is getting reduced during influenza infection, but at the same time, in the animals which are infected with SARS-CoV, there has been increase in virus. Similarly, in case of chromiatails, we don't see any variation, and enterobacterials also there is no significant variation. So like that, different infections showed different kind of abundance, and these are the bacterial orders which are getting reduced during SARS-CoV-2, which are increased during but influenza virus. But it can see there is a clear changes in the. So you can see in this case of lactobacillus, which we all known as a good probiotic and a very symbiotic bacteria. You can see on comparison to during influenza, the lactobacillus level is increasing, but during COVID-2 infection, it's getting reduced. So it shows that commensal bacteria are getting reduced. And also, as I mentioned, they have collected the nasal washes from the nasal wash. Is just they added some PBS into the nostrils of the animals, and then they recollected. Okay, so you can see as the days goes on, you can see these bacterial orders like Bacillus, Lactobacillus, Pseudomonas are getting falling down, and there has been a group of bacteria which are getting increased, including Enterobacteriales, Sphingomonas, and these are largely have most of the pathogenic bacteria like Streptococcus, and those bacteria are present. So you can see there has been a significant, a slow but a significant increase in the bacterial orders. So these are some of the key inferences. So on comparison to influenza A virus infection, there is relatively less changes in the bacterial diversity at the phyla level. And we have observed an inverse correlation in disturbance of microbiota with the species, the bacterial orders which are reported to be increased during influenza, decreased during SARS-CoV, and vice versa. So, and also among some of the, we have observed blooming of pseudomonodales, which is not present in, but when you see at the day 7, there is a reduction in the pseudomonodales in case of SARS-CoV, and some good commensal bacteria like lactobacillus seems to be significantly reduced on comparison to influenza. So these may play a major role in the COVID-19 outcome. So similarly, we have collected some sample data from humans initially reported. Actually, this sequencing has been performed initially only. Now, as they know the complete RNA sequence, now only qPCR is performed. And another thing about these viruses are they are RNA viruses and they are not retrovirus. Okay, they don't, just like in case of HIV, they do a reverse transcription and they will integrate into the genome. But these RNA viruses, they have RNA dependent RNA polymerase, so they will replicate in the cytoplasm itself. Okay, so initially to identify the sequence, a lot of RNA sequencing has been done. So based on that, we have collected data from initially infected patients in Wuhan, China, and we have seen a similar pattern of here we don't have a comparison with the influenza, just we have compared the control and the infected humans. So we have seen a similar pattern of no big change at the bacterial levels, and these are some of the species which are known. So this influenza data we have gathered from a different publication, and we have seen some species which are known to be getting more abundant during SARS-CoV. So there are some major limitations of this study because in both the cases, whether it's a human samples or ferrets, we not directly did the experiment. So we lack a lot of clinical details, and also similarly we don't know the output of the humans which we have studied. So irrespective of these limitations, we made some interim conclusions that there are changes in the upper respiratory tract as being made by the during the SARS-CoV-2 infection, which seems to be happened progressively. And we have also seen, so now we are working on this. We have applied for the grants and we are waiting for the permission from the government of India to work on what is the variation on. We have been struggling to get this grant sanction for the more than for the past 8 months. Okay, so in parallel we have now applied for a Indo-UK grant. We have had a collaboration with Imperial College London, and another thing that I want to share with you is, as you have heard in the news, the mortality rate in India is relatively low when you compare to European countries. That we all know. But the other unknown thing is the people who are getting died due to COVID-19 infection in UK or in European countries are largely South Asians who belongs to SAARC nations like Indians or the major people who are getting died due to. So there has been a call from the UK Indo-UK project to understand what causes Indians living in India getting recovered from COVID, but the Indians who have the same genetic background living in UK are getting ending with high mortality. So we have, in our collaboration, we have proposed on studying the microbiota of both the gut microbiota and nasal microbiota of people, Indians living in India and as well as the Indians living in UK. And probably by next month, and we will know the result whether we got this sanctioned or not. But in parallel we are trying to work with how there has been changes and whether this has a role in this COVID-19 output. So by understanding this, we can devise new therapeutic strategies. So vaccines is other way of preventing the disease, but still for RNA virus, a vaccine is not a big hope. There has been a lot of news, and we have seen the trials has been started even in India. But for RNA virus, it undergoes a lot of mutations. So how much a vaccine will stay for years is a big question. Even for influenza virus, there is no standard virus vaccine. Even for HIV, still there is no virus. So even for COVID-19, we are not sure how much these vaccines will be hopeful. But the trials has been going on. But on the other hand, how by understanding this microbiota can help in fighting against COVID-19. So the one way is by understanding if there is as I said, any antibiotic or these things will kill everybody. So we can target some specific bacteria, specific antibiotics, or some strategies can be developed to kill the secondary bacterial infection. And we have seen commensal bacteria, so good bacteria are getting reduced. So some strategies can be developed to provide them as a nasal probiotics to help to recover the healthy ecosystem. So these are the aspects which we have discussed on how microbiota will play a role on SARS-CoV-2 infection. So as we see, everything is double-edged. So there is another important thing which is a big worry for us, which is not being discussed or which is not being thought by many people. Okay, so what is the effect of disinfectants on microbiota? So when we speak about biodiversity, there in the recent years there has been a lot of awareness on biodiversity, climate change. Even there are hypotheses this virus has become a pandemic and infectious to humans due to destruction of forest and the animal habitats. So on the similar way, there is a huge microbial diversity present all over the world, starting from forest, wild animals, and everywhere. And in the name of defense against this COVID-19, we are using a lot of disinfectants, right? Everywhere, even that has been made as a mandatory rule, and it has been followed everywhere. So right from the education institutions, even during travel. And so without knowing what it causing, we are using a lot of disinfectants. And on the other hand, while we are washing our hands, and after that we are releasing into the soil and to the environment also. So as I said, not all, for example, this is the mechanism by which the soap molecules kills the bacteria or any other microbes, viruses. So this SARS-CoV also has a lipid layer around us, so this by a simple polar bipolar mechanism it kills. So but it not kills only the SARS-CoV, it kills everybody. Okay. And most of the some of the antibacterial soaps or the sanitizers which are present in the market has these compounds like triclosan and tricloban, which has big they are known to present carcinogenic activity which can cause cancer, and they cause endocrine disruption. They are allergens and they are getting released into the environment, and the fish are one of the major uh, reservoirs of where the toxicants gets bioaccumulated. So in another way, again going to cause a big environmental disruption. And when we come to the microbiota, the sanitizers which are using day to day, right? As I said, the microbes are present everywhere in our human body, and the skin is one of the major reservoir of the microbiota. So you can see there is a big variation on the type of bacteria which is present in different portions of our hand. If you take the hand, it has a different group of bacteria present, while when you take the forearm, different groups of bacteria are present. So as I said, they are not simply presenting; they are playing a key role. Okay, and there has been a lot of studies which showed that skin microbes plays a major role in our immune system. Okay, so it produces, so by destroying in the name of defense against COVID-19, we are destroying our microbes which are present in our skin itself. So what is their impact in the future in our immunity is a big question which needs to be studied, and a lot of we have to use at today's situation we don't have any other option, but still we have to think on how to there some strategies, some studies are needed to understand how much disinfectants are released into the water bodies and how to uh, bioremediate them, how to remove them similarly into the soil, in food substances, and yeah, as well as in the human body, how much microbes we have lost and how we can recover them, like this are needed. So the other one more problem is antimicrobial resistance. So in the last one half century, a lot of antibiotics has been used. Even we people, just when we get a cold or even during this COVID, lot of antibiotics are being used. Okay, and when we use all these hand sanitizers and those things, as I said, not all the microbes are getting died. Some microbes will start developing resistance. As a result, there will be a flourishment of drug-resistant bacteria, which is a major problem today with tuberculosis and other infectious diseases. So we have used a lot of antimicrobials today, and these may lead to new drug-resistant bacteria, staphylococcus, or any other mycobacterium tuberculosis infection. So all these are the new challenges during this after this uh, uncontrolled use of sanitizers and disinfectants we are going to face. So this is what I am propagating nowadays. So making sterile may be clean but cannot be called as healthy. So making nothing, எதுவும் இல்லாமல் செய்வது சுத்தம் எனலாம் ஆனால் ஆரோக்கியம் அல்ல. So but today we have no other choice. So I'm not advising you people to not use sanitizer or not to wash hands, but studies are needed how these things can be controlled and what are the implications in the future. And this is another example how we are destroying the environment. Now we are using the face mask and everything. It is being released into the environment everywhere. There has been a lot of reports, even the mask and syringes and medical waste especially during this COVID-19 pandemic has been detected in deep oceans, even in the areas which are not habitated by human beings. So all this will have a profound effect on the environmental microbiota, which in turn will have a different effect on the soil, fertility, climate change, and everywhere. So these are the different projects which we are. So my lab is termed as Hemomicrobiomics Laboratory. So the major aim of our lab is to understand how bacteria interact with the different chemicals and what is their impact on environment and human health. Okay, so this is the final message that I want to deliver, especially during this COVID-19 situation. So we are not all bad. I am just speaking on behalf of the microbes. We are not all bad. Okay, as shows in the advertisements, if you kill 99.9% of the microbes, there will be no Earth. Okay, because the microbes provide oxygen and they control the climate change. They increase the soil fertility and they provide us the food. They provide immunity to us and good health to humans. They created the soil, mountains, and minerals and everything in this world. So if you have, I'm happy to share my thoughts with the students. So if anybody is interested, you can contact me through this email ID. So I would like to thank the institution for providing me this position and the president of KMCH Research Foundation, clinicians, collaborators, and students and my funding agencies. So thank you all. If there are any questions, I'm happy to answer.
Thank you, sir. Students, do you have any questions? You may raise your questions. Dr. Velmurugan is there to clarify. Yes.
Okay.
Thank you, Dr. Velmurugan. I would like to ask you one question. Is there any relation 100 years back Spanish flu was there? No? Spanish flu.
Sorry, sir. Sorry, your audio is not clear, sir.
Spanish flu 100 years back. Spanish flu. Yeah?
Spanish flu, yes.
Okay. So now COVID-19. COVID-19. Okay. So is there any scientific changes like that, which is just to balance the ecosystem like that, the disease like that is coming like that? What is your opinion? The relationship of...
I'm not sure why it's exactly 100 years, once in 100 years. Maybe from a scientific point of view, it should be considered. But I don't have exact hypothesis to say exactly why it happens once in 100 years, such a big epidemic is happening. But this time it's largely due to one of the reasons why it's become a global pandemic now in comparison to Spanish flu. Now the world has become very small due to increased transportation and other facilities. Maybe that's one of the reasons it has widely spread on comparison to 100 years back. Because when Spanish flu happened, maybe the villages in Madurai or even the people who are living in Indian cities will be unaware of the pandemic.
Yes. Okay, thank you.
Students, do you have any queries or questions? Be free to raise your questions.
So maybe I will just type my mail ID in the chat box. So students have any, they can contact me by email if they want any clarification or they want to know more about the work.
Definitely, sir. They might have some hesitation to raise the question in the group. We might ask you in person.
Okay.
Thank you, sir. Thank you for your scintillating presentation. The one thing we have been so astonished is that making sterile weekly but not healthy. This is what everyone knows, but we are not being used to it. We are using the disinfectants now and then continuously, though it is negative effects. Sir, it was a terrific presentation. We are pleased with your informative speech. Thank you, sir. It is really a point to us when we come to know that your first PhD student is from our department. That itself is a great motivation to us. Yes, you maintain a remarkable point in the face of a potentially hostile audience. Thank you, sir. Thank you once again. Now, we are into the next session. It is purely for the Students, here we go over the e-poster presentation. Students, this is your arena to exhibit your talent. So today we have four students to present their e-posters. Virtual seminar run by technology in human welfare. The e-poster competition is to be resumed. The first participant who is here to present his e-poster is Deepshika, third BSc by technology. Deepshika, are you ready?
Yes, ma'am.
Yes,
Ready,
you
ma'am.
can start with the presentation. This presentation will be judged by our speaker, Dr. G. Velmurugan, and our coordinators are. Okay, so your presentation will be up to 3 to 5 minutes. For the first 3 minutes, you have to present it, and the next 2 minutes, you have to answer the queries raised by our judges. Right? Deepshika, your time starts now.
Ma'am, shall I start, ma'am?
Yeah, all right.
Okay, Dr. Velmurugan needs some prerequisites. We'll meet in next 10 minutes. You be on time.
இப்போ வந்து அந்த என்ன சாருடைய நல்லதுன்னு தான் போட்டுருங்க, அப்போ சரியா கேட்கல. அது பிரின்சிபல் சார் இருக்கு. நான் ஷேர் தான் பண்ணி விட்டேன் கூகுள் டிரைவ்ல இருந்து. அவங்க உங்கள்கிட்ட இருந்தா கூட அந்த கூகுள் டிரைவ் அட்ரஸ்ல இருந்து ஷேர் பண்ணுவாங்க. இல்லை நான் இப்போ பிரின்சிபல் சார் எடுத்துக்கிட்டேன், என்ன பேசுறேன் அந்த மெயின் டைம்ல அவங்க இதுல தான் குறகுறன்னு இருக்குது, அவங்க இதுல ஷேர் பண்ணுவாங்க.
ஸ்பீக்கர்ல நம்ம மொபைல்ல போட்டு அது நல்லா வருது. அவர் என்ன சொல்றாரு அசோக் சார், இதுல இருந்து நம்ம கூகுள் மீட்ல போட்டா அந்த ஆடியோ கொஞ்சம் குறையதான் செய்யும்.
dynasty with Madurai as its capital was known to the ancient Greeks. Madurai appears on Ptolemy's map of the world. It was considered by foreigners as Athens of the south, known as Temple City. It is a sacred center or a cosmic center. It is ruled by goddess Meenakshi. Under the dynasty of goddess Meenakshi stands like a towering titan between centuries, an academic institution, the Mijro College. The Mijro College has a hoary past spanning a century and a quarter. It has served the cause of higher education with dedication all these decades. Department of Biotechnology was established in the prestigious institution, the Mijro College, as a self-supported course in the year 2008 to prepare the students for this competitive field of science. The curriculum and syllabi is crafted to impart current advancement and understanding about biotechnology. The department works towards a vision to produce disciplined and competent students of high caliber to become socially committed and ethically strong to meet the challenges. To achieve that goal, the department mission moves in a path such as fostering excellence, adopting proper pedagogical methods to ensure quality education and academic excellence, and providing a conducive environment for learning, following ethical, moral, and spiritual values. The curriculum is designed to meet the demand for evolving developments for students who be competent to pursue higher education, acquire skills for career opportunities in various biotechnological industries. Upon completion of the course, students would have a thorough knowledge of biology. They would acquire the ability to work on their own or in group, and not the less good analytical thinking skills, and be able to use a computer and the main software packages competently. The department has a well-equipped laboratory with instruments such as centrifuge, spectrophotometer, chambers, electrophoresis units, deep freezers, etc. The department library has a wide collection of books of varied titles that numbers to 1,600, and also books of various competitive exams. The classrooms are spacious with sufficient lights and aeration facility. The salient feature of the department includes enhanced learning opportunities, ability to match students' holistic needs. CBCS and OBE will facilitate vertical integration of knowledge and holistic development of self. Continuous internal evaluation. A teacher offers a course assess performance of the students. All appropriate steps are taken to create and maintain infrastructure required for quality teaching and learning. Psychological counseling and guidance services through mentor system. Mental health is critically important to teaching learning processes, creativity and congenial environment in academic institution. This is not the end. There is a lot to learn, textbook to achieve.
It is really wonderful to watch the lenses of the biotechnology. Really, the credit goes to Harikaran, third BSc Biotechnology, who has crafted this video, and also the voice been read by rendition has been done by Ratnashri, second BSc Biotechnology. Hats off to you, the boys and girls. So definitely you can, you have proved this, you can do anything if opportunity strikes. So we'll just continue with the e-poster presentation. First, it is over to Deepshika. Deepshika, are you online?
Yes, ma'am.
Okay, you can start with your presentation.
Good morning to everybody. I am Deepshika, third biotechnology. I am here. I am going to present my poster that is about drug delivery by nanoparticles. Drug delivery by nanoparticles is an emerging technology. If you want to know the need of the technology in current situation, we have to see the defect of the current medication techniques. Current medication techniques involve the usage of pills for any diseases to be cured. If diseases want to cure, then the pill should reach the specific site of infection and do its action. If it doesn't reaches the specific site or only a small amount of the drug reaches the specific site, it won't be effective against that disease. On the other hand, if the pill reaches the site other than the infected site, it leads to the side effects. But in case of nanotechnology, it differs. The mechanism of drug delivery in both the cases is same. The size matters. In case of drug delivery by nanoparticles, drugs are carried by nanoparticles. Nanoparticles are ultra fine particles which are usually sizes about 1 to 100 nanometer, as they are very small in size and also they can carry a large payload of drug. Nanoparticles act like a vehicle. A truck exactly means a biotruck. The biotruck was loaded with the drug and injected into one's body. When it reaches the specific targeted site, it releases the content of the drug. Now we see about the mechanism of the drug delivery. The mechanism doesn't difference in both current medication system. It means the pill and the nanoparticle delivery. If you want to cure any disease, the drug should reach the specific site. Before that, it has to cross the 5 barriers in our body. The mechanism is a delicate mechanism, but we can see in a short manner. The first barrier is our stomach. When we take up the pill, it first reaches the esophagus. Then it goes to the stomach. That acidic environment of the stomach can easily degrade our drug in case of pills. But in case of nanoparticles, it doesn't affect any of that. Then approximately after 37 minutes, the pill enter into the intestine. Intestinal wall was so small, so some of the portion of the pill has to degrade, and because it should be chemically fit. But in case of nanoparticles, as they are already small, it doesn't affect any of this. Then the pill reaches the barrier liver. Liver has many enzymes. Even the pill was degraded in this place. But nanoparticles didn't affected by this barrier. The next barrier is bloodstream. After passing through this, it reaches the final barrier, the blood-brain barrier. The blood-brain barrier allows only a small sized particles to pass through. If our pills were very large in size, it cannot pass through and it wouldn't be effective against the disease. But in case of nanoparticles, they are very small and can easily cross through the blood-brain barrier and can reach the targeted site easily. The other use of the nanoparticles is it can easily identify the healthy cell from the diseased cell. In case of diseased cell, it produces certain chemicals on the surface. So the nanoparticles can easily identify the difference between the healthy cell and a diseased cell. Nanoparticles have the potential to improve the stability and solubility of the cargoes, I mean the drugs. It promotes the transport across the membrane and also prolongs circulation times to increase the safety and efficacy. Nanoparticle research has been widespread, generating promising results in both in vitro and in small animal models. The nanoparticles are made up of many compounds. Now we see about the nanoparticle classes. The first class was lipid-based nanoparticles. The lipid-based nanoparticles are spherical in shape. Uh, it comprises of the drug, and the drug was surrounded by one lipid bilayer, and the lipid bilayer was surrounded by the aqueous compartment. As a delivery system, lipid-based nanoparticles have its own advantages. The advantages include its self-assembly, its biocompatibility, and its high bioavailability. It also have the high ability to carry a large payload of drugs. Now we see some of the lipid-based nanodrugs. Doxil is a nanodrug which is used to cure ovarian cancer and multiple myeloma. Ambisome is another lipid-based drug which is used to cure fungal and protozoan infection. And Markibo, it's also a lipid-based nanodrug. It also cures lymphoblastic leukemia. Then the other class of the nanoparticle was polymeric nanoparticles. Polymeric nanoparticles can be synthesized from natural or synthetic polymer or even from a pre-formed polymer. Generally, they act as a good delivery vehicle because they are biocompatible and have simple formulation techniques. Polymeric nanoparticles can be synthesized using techniques such as emulsification and nanoprecipitation. In this case also, the drug was encapsulated within a polymeric nanoparticle core, which is entrapped in a polymer matrix. The mechanism was same in all the cases. This enables the delivery of various payload including hydrophilic or hydrophobic drugs, even proteins and vaccines. Now we see about some of the polymer-based nanoparticle drugs. Copaxone is a polymeric nanoparticle drug which is used to cure multiple sclerosis. Eligard is also another polymer-based nanoparticle drug which is used to cure prostate cancer. Adenovate is another drug which is used to cure hemophilia. Then the next class of the nanoparticle was inorganic nanoparticles. Inorganic materials such as gold, iron, and silica are being used to synthesize a nanostructured nanoparticles. They are also used in drug delivery. These inorganic nanoparticles are precisely formulated and can be engineered to a wide variety of applications. I- The inorganic materials have unique physical, electrical, and magnetic properties of their own. The important inorganic nanoparticle was AUNP, which is gold nanoparticles. They can be easily functionalized and additional properties and also have their own delivery capabilities. Some of the inorganic based nanoparticle was dexferrum. It is used to cure iron deficient anemia. The another one was venofer. It is used to cure iron deficiency in kidney diseases. Here, there are several advantages when we compare to the current medication system, the pills. But in case the number of nanomedicine available to patient nowadays is very drastically low for the field because of translational gap between animal and humans. Yet, the upcoming research will overcome everything. The difference across species limits the behavior and functionality of nanomedicine in human. Hence, the research is going on towards its success. As this is an emerging technology, we are less known about its miracle. When the research reaches its final successful step, we can promise a disease-free world. We can eradicate the disease completely from the world, but we can protect the humans from affecting the disease. Thank you.
Thank you, Deepshika. So you have concluded that biotechnology is working towards a disease-free world. It's good to hear. Sir, do you have any questions, sir?
What's the current scenario about the nanoparticle drug delivery?
Deepshika?
It's not.
So what is the current scenario of the nanoparticles as a drug?
Sir, ma'am, the research is going on, ma'am. Ma'am, Doxil is successfully treated for ovarian cancer, ma'am, nowadays. In America, it is widely used to treat the cancer, ma'am, mainly ovarian cancer, ma'am.
Here, sir. Dr. Velmurugan, sir. Dr. Velmurugan.
Line இருக்குங்க.
Okay.
ஆர்த்தி வந்து சரி அந்த
The next presentation. Submission. Submission parents.
Yes, ma'am. Good morning, ma'am.
Yeah. You can start with the presentation. Just wait.
அவளை பிரச்சனை பண்ணலாம்.
அவரு வேணாம் சொல்லிடுங்க சார் அப்புறம்
யார் வர்றா?
Somebody, you just presented orally.
You did not go with your e-poster presentation. You just have a look into the presentation which we are sharing.
Your screen question.
Somebody can stay there as well. We are sharing the presentation. You stop presenting it.
Ma'am,
Then just stop.
it's not clear, ma'am.
It's not clear, no? நீங்க பின் பண்ணுங்க.
சபரிஷன் அதுல போட்டுருக்கு. ஆமா நீங்க சொல்லுங்க. சபரிஷன் தர
Sir, it's not clear audio, sir. Hello?
என்னது bro?
Hello?
Audio clear இல்லைங்கற.
Sir,
ரெண்டு பேர் போன் இருக்கு யாரா ஒருத்தர். இப்போ நீங்க எதுவும் பண்ண வேணாம். நீங்க உங்களுக்கு நாங்க பிரசன்ட் பண்ணுவோம். அப்ப உங்களுடைய போஸ்ட் வந்து நாங்க டிஸ்ப்ளே பண்ணுவோம். நீங்க வந்து அத பார்த்து நீங்க பிரசன்ட் பண்ணா போதும். நீங்க என்ன போஸ்டர் நீங்க மெயில் பண்ணீங்களோ அந்த போஸ்டர் இப்ப நாங்க டிஸ்ப்ளே பண்ணுவோம்.
okay, sir. Okay, sir.
ஓகேவா? நீங்க வந்து அந்த பிரசன்டேஷன்ல பார்த்து நீங்க சொன்னா போதும்.
Okay, sir.
நீங்க வேற எந்த பிரசன்ட் ஸ்கிரீன் எதுவுமே கொடுக்கறது.
Okay, sir.
இப்போ உங்களுக்கு ஸ்கிரீன் தெரியுதா உங்க பிரசன்டேஷன்? I mean உங்க போஸ்ட்
Yes,
தெரியுதா?
sir. Yes, sir.
Okay.
Submission paid sir. We can start the presentation.
Okay. Good morning, everyone. First, I thank Department of Biotechnology for this opportunity. Let's go through the presentation. Leukocytes in the blood have to be removed before the transfusion in order to avoid blood-borne diseases and some side effects. What are all the side effects we can facing? Allow immunization to histocompatibility antigen, which means allow immunization immune response to the foreign antigen encountered through to cells or tissue from genetically different member of the same species. White blood cells may suppress the recipient immune system. We already know that. So, leukocytes provides no benefit to the recipient but can carry bacteria and viruses to the recipient, which may lead or cause to some diseases. And viruses like cell-associated viruses like cytomegaloviruses, CMV, Epstein-Barr viruses, and T-cell T cells lymphotric viruses or some viruses which may cell associated viruses goes through by the WBC, which means leukocytes through donor to recipient, and immunosuppression and graft versus host diseases, which are the diseases we are facing by blood transfusion. These effects are because of WBC, so that reducing WBC may prevent these harmful effects. So there are many some techniques to reduce WBC. Let's see the techniques involved in WBC reduction. There are 5 basic techniques. They are differential centrifugation, sedimentation, cell washing, freezing, and thawing, and filtration. First, differential centrifugation. In that blood packs are centrifuged, blood cells will sediment according to their specific size and density. So we already know that the centrifugation separate the cells, separate the particles, depend on the size and the density. So we can separate the RCB, WBC, and cell-free plasma separately. This undergoes the second centrifugation after the addition of an isotonic saline solution to the packed red blood cells. And next, sedimentation. Without the use of centrifugation, sedimentation can be enhanced by the addition of agents that promotes the formation of RBC aggregates known as rheolax formation. Dextran sedimentation, which is low cost, but it took some more time to separate. Next one is cell washing. RBC washing combines differential centrifugation and continuous dilution of the cells using isotonic saline solution. And this is also a next freezing and thawing. This process developed for long-term preservation of RBC. To protect them against freezing, a cryoprotective agent, for example, glycerol, is taken up by the RCB, and freezing ice crystals that rupture the cell membranes during thawing are formed in the leukocyte. In this process, the WBC's residues are remaining in the process. So this process is not longer for the use. Next one is filtration. Filtration is the best yes, of course. Filtration gives more efficient to the separate the RBC. And these are development of leucofilters. The first described in 1926 by Fleming by cotton wool. That cotton wool separates the WBC and RBC by the filter process. In subsequent years, different materials were used, like Orlon, Dacron, Teflon, nylon fibers, and those among these nylon fibers are giving more effective. And it is improved in 1972 by Dieffenhorst by tightly packed cotton, and it gives a result of 95% WBC were removed with the loss of 10% RCB. And upcoming years, after 2 years, the disposable leukocyte filter introduced with the polyester fiber. It is the most effective thing up to date, which removed 99.9% WBC, and recent and developed filters are removing mostly 99.999% WBCs. There is a mechanism involving in a filter. They are cell sieving, pore size effect, margination effect, complement action, cell addition, surface chemistry, surface charge, surface wettability, and many more. Now I am going to explain on two mechanisms. One is pore effect and complement activation. Pore effect, we already knows that the size of the RBC and WBC. The size of the RBC is 7.5 to 8.7 micrometer in diameter, and 1.7 to 2.2 micrometer in thickness, and WBC 12 to 17 micrometer in diameter. So that we can conclude that RBC is smaller than WBC. So that the filter has filtered the smaller particle. It allows the smaller particles down and fix the larger particles in the trap, like trap-like structure. And the complement activation. Complement activation is also known as to be involved in the addition of leukocyte to artificial surface. Artificially, we are giving the filter, which will be the substrate will addition, which is attached with the substrate, with this artificial substrate, and we can activate the addition in the cells. How means activation of calcium ion and magnesium ion generate C3A and C5A fragments, which are which are in cell membranes, which will, when it is increased, it will very adhesive to the um, artificial surface. Next, pros and cons. I mean, those are the pros. Clinical benefits of leukocyte reduction, proven benefit relevant clinically, reduced frequency and severity of FNHTRS, reduced risk of CMV. Most of the not most of the cell associated virus are removed by this process. Probably clinically relevant. Yes. Reduced organ dysfunction when it is immunosuppressor and graft versus host disease were mostly rejected, and transfusion and transmissible of bacteria, and unproven clinically, reduce the risk of GVD, GVHD, and reduce the risk of TRAL. And we can conclude that conclusion from this stage. Now the currently available leukocyte filters more and more effective, uh, and and it is 99% the leukocyte from whole blood reducing, whereas the blood cell loss only very small. Although current filters performs are acceptable, optimization is still desirable. Thank you.
Thank you, Peter, sir. Sir, do you have any questions?
So, thank you, Mr. Paral, sir.
Thank you, sir.
ஓகே, இன்னைக்கு பிளட் பேங்க்ல பிளட்லாம் வச்சுக்காங்களா, அது ஹவு லாங் அந்த பிளட் பேங்க்லயே வச்சிருப்பாங்கன்னு தெரியுமா? பிஃபோர் யூசிங் டு டோனர்.
கிளியரா கேட்கல சார், சாரி சார்.
Presently, the blood in the blood bank, the bloods
Okay,
are being
ma'am.
stored. So, do you have any idea how long it can be stored in the blood bank?
Ma'am, yes, ma'am. Blood bank.
42 days.
Ma'am, in India, ma'am, this process, in the process வந்து ரீசெண்டா இப்ப வெரி ஸ்மால் கண்ட்ரிஸ், அதாவது வெரி ஃபியூ கண்ட்ரிஸ்ல தான் மேம் பண்ணிக்கிட்டு இருக்காங்க. அதாவது இந்த WBC வந்து செப்பரேட் பண்றது. இது வந்து
not asking about this technique, pa.
Ah.
How long a blood can be stored normally? A blood can be stored in a blood bank.
Oh, 6 months, ma'am.
42 days.
Its expiry date is 42 days.
Yes, you say 6 months, the other one says 42 days. Be clear.
Okay, ma'am. 42 days, ma'am.
Just for 42 days.
Yes, ma'am.
Okay, thank you, parents and Sabari. It was a wonderful presentation. So we'll move on to the third presentation.
Thank you, ma'am. Thank you, ma'am.
Thank you.
Thank you, ma'am.
Third presentation. Jyoti Priya, are you ready?
Yes, ma'am.
Just wait.
Shall I start now? Ma'am?
Jyoti Priya,
Yes,
you
ma'am.
can start with your presentation.
Okay. Good morning to all. I am S. Jyoti Priya from 3rd BSc Biotechnology. And my post is about one of the successful and important pharmaceutical product of marine biotechnology, that is ziconotide. Ziconotide is a painkiller which is obtained from the venom of a marine snail, Conus magus. It is not a natural form, it is a synthetic form of omega conotoxin peptide which is present in the cone snail. Conotoxin means it is one of the group of neurotoxic peptides which differs species to species. And the ziconotide was discovered in the early 1980s by the scientist Michael McIntosh, and it was developed into an artificially manufactured drug by Irish Elan Corporation. And it was also approved for the sale under the brand name Trialt in 2005. This drug was mainly used by the patients having a chronic pain, severe chronic pain, that is prolonged pain caused by AIDS, cancer, or neuropathic pain. The main mechanism of this painkiller is it blocks the N-type voltage-gated calcium channel. This calcium channel present in the nerve terminals, dendrites, and also in the neuroendocrine cells. The main function of this calcium channel is to release neurotransmitters, and it also involves in the pain pathways. So this blockage action inhibits the release of neurotransmitters, and it blocks the pain signal, which results in the pain relief. And the ziconotide is also called as intrathecal ziconotide because of its administration route. Intrathecal means administration of drug into the spinal canal. And the therapeutic use of this ziconotide is, it is also suggested for the patients with certain pre-existing mental disorders like psychosis, and it has an excellent safety profile with no neurotoxicity, no mutagenicity, and no inflammation, no teratogenicity. Teratogenicity, the word teratology means that it is a study of abnormalities, so no teratogenicity and low immunogenicity. It also has some side effects. If this drug is overdosed, they may cause with trouble in breathing and also in meningitis. The future prospect of the ziconotide is the omega conotoxin peptide. This is a biochemical or under research to develop a drug to treat a brain injury. And ziconotide offers some deep understanding regarding the future development of new analgesic drugs, which has the same mechanism of ziconotide, but might improve safety and tolerability. And I conclude that ziconotide is a powerful analgesic, not only for reducing pain, but also improves the neurological functions. And the applications of marine biotechnology not only include pharmaceutical products, it also include cosmetics, improving the production of marine organisms, and also in the production of novel products such as food and feed products. Thank you.
Thank you. I'd like to ask a simple question. Is there any relationship between mutagenicity and carcinogenicity?
So both are same, sir. Mutagenicity is sudden change, and the carcinogenicity is also its which forms cancer cells.
Okay. I am asking only what is the relationship?
Cause cancers.
Okay. What about mutation? What about mutation? I'm asking because most of the studies shows that 70% of the mutations may lead to cause a carcinogenic or carcinogenicity. Okay. Thank you.
Thank you, sir.
Thank you, Jyotipriya. Thank you for your wonderful presentation. Next presentation is by Vishnu Kumar, second BSc Biotechnology. Vishnu Kumar, are you ready?
Yes, ma'am.
Vishnu Kumar,
Ma'am?
you can start with your presentation.
Yes, ma'am. I'm present as well, ma'am.
Yes, Lee. Vishnu,
Yes, ma'am.
you can start with
I'll
your
start.
presentation.
Yes, ma'am. Hello, everyone. Good morning. I am Vishnu Kumar, Department of Biotechnology. Today, the topic of my presentation is about how mRNA 1273 vaccine works. What is the vaccine? A substance used to stimulate the production of antibodies and provide immunity against the pathogen. Provide immunity against pathogen, and there is two main types of vaccine: is attenuated and inactivated. The mRNA vaccine is attenuated vaccine. What is attenuated vaccine? Attenuated vaccine is created by reducing the virulence of pathogen, but it's viable and it is stimulated strong and effective immune response. And mRNA vaccine have elicited potential immunity against infection disease target. This mRNA 1273 vaccine is given to intramuscular injection. It codes for infused the S protein, which is spike protein of coronavirus. In this vaccine, the mRNA is encapsulated by lipid nanoparticle. After they inject the vaccine, particle bump into the cell and fuse them, and the particle release the mRNA into cells. The mRNA is translated to protein, which is S protein. The vaccinated cells also break some of the proteins into fragments which they present on their surface, and protein and S proteins can recognized by immune cells. When the vaccinated cells are lysed, the debris will contain S proteins, which is taken up by immune cells called APC, which is antigen presenting cells. When other cells called cytotoxic T cells, which can be help immune cells to fight the infection. And one of the immune cells, B cells, some B cells are able to lock the spike proteins. And if the B cells are activated by cytotoxic T cells, they will pour antibodies to target the spike proteins, and the antibodies latch on the S proteins and prevent fusion. The APC can also activate another type of cytotoxic T cells to destroy coronavirus infected cells. And I will conclude the mRNA 1273 vaccine is 94.5% efficacy in preventing symptomatic COVID-19, according to the phase 3 results reported in New England Journal of Medicine. Thank you for all listening my presentation.
Thank you, Vishnu Kumar. Sir,
Hello, Vishnu.
Sir, yes, sir.
I have one question. How, because mRNA is highly unstable, so how
Yes,
it
sir.
is delivered into
Sir,
the
is
cell?
sir, the lipid nanoparticle வந்து பைண்ட் ஆயிருக்கு சார்.
Okay. So lipid nanoparticle, it is encapsulated in lipid nanoparticle.
Yes, sir, encapsulated.
Okay. Thank you.
thank you, Vishnu,
Thank you,
for your
sir.
wonderful presentation.
Thank
The
you, ma'am.
results of this presentation, along with tomorrow's presentation, will be announced positively on Wednesday. Last but not the least, it is a courtesy to thank everyone. I call upon Dr. Arul Muthukumar Kumaran to deliver his vote of thanks.
Thank you, ma'am. So good afternoon, all. I hope this was a wonderful session. that had a guest lecture by a speaker on a modern topic, and it was also a new experience for the students to present their posters. So I hope that you enjoyed. And now it's a time for thanking the people. So I will go by. So I made a slide. So we have to thank so many, since it is our maiden attempt to make an e-seminar. So first of all, I would like to thank our Mizora College board. So they encourage us by funding every year to organize such seminars. We thank our secretary and treasurer, who also constantly encourage us and by their visits and also by their words. And we have a special thanks to our principal, Dr. J. Suresh, and we approached him to give an inaugural address. Even he was busy, he agreed and made a wonderful inaugural address within a short span of time. So thank you very much, sir. And of course, so our KMR sir, he used to encourage us continuously. So if he was here now, he would have encouraged and he would have joined us. So thank you, sir. Then we have to thank our coordinator, Dr. N. Panirsan. So we have a wonderful coordinator who organizes us very well. And when we approached him with the idea of an e-seminar along with the student presentation, so he welcomed it, he immediately accepted, and he was involved in and asking the principal for inaugural address and getting this computer room. And he sat with ourselves, and he is even now sitting and asking questions. So thank you very much, sir, for encouraging the students as well as us. Then we want thanks. So without the work of our department colleagues from biotechnology, Ms. Suguna, Dr. Vimal, Dr. Kritika, and Dr. Bhaskaran, this event would not have been possible. And Bhaskaran made a wonderful design of the poster. What you see is it was made by Bhaskaran sir, and it was a total effort. They made a contribution in each and every part, and it was a very good one. Of course, tomorrow you have one more session, so let us see. So thank you very much for our department colleagues. Then again, there was also much support which came from department colleagues from our Department of Microbiology. So thank you so much. And then Mr. Ashok Kumar from Department of Computer Science, Assistant Professor. So he was so instrumental in making arrangements for this e-seminar in Mohan Hall of our Mizora College. So without his help, we would not have possible to host this seminar. So thank you, Ashok. Then of course, we have to thank our non-teaching staff and lab assistants. So their help was also there, whatever would be minimum or maximum. So we have to thank them. First of all, I would like to thank Mr. Jayakumar, lab technical assistant in Department of Biotechnology, and Dasanna from Department of Microbiology, and Mrs. Meenakshi for their routine helps. And special thanks to Mr. Hariharan, who made the video for both KMR sir and as well as department video. Ms. Ratnasri, who made her contribution by voicing, and Mr. Aswin for that image of Dr. KMR. Then it's a time to thank our speaker, Dr. G. Velmurugan. So he also accepted accepted our invitation and gave a wonderful talk. It was I hope it was a very inspirational talk and informational talk also. Dr. G. Velmurugan, he was my junior during my MSc, and he was an energetic student. So, I want to tell a few incidents about him that would be motivating. So when he did a PhD in Madurai Kamaraj University, and before that he worked in different places. He went to Germany, and after finishing PhD, he worked for some time in IIT. Then again he went to US for 1 year. Then he came back, and at his age, he is an independent researcher. So being an independent researcher itself is a big credit. So you can do your own research, but you have to drive funds from government companies. So at his age, he has attracted a fund of 1.2 crores from the government. That is not a joke. And he has 3 more funds worth more than 50 lakhs. And when he was working in the lab, he had a temporary health problem. So he was not allowed to work in the lab after 7. So what he did was he used to come he did not stop working. He used to come at 4 o'clock and work till 6. And he was a voracious worker. and he's very conscious about the environment. So whatever research he does, it will have an impact on that will have impact on society or environment. So very nice of him for presentation. So you can contact him for further if you have any doubt, or after completing MSc, if you want to approach for a PhD, you can mail to him. So he is free. He is student friendly. And last but not least, so without the participation of the students, UG and PG students from Department of Biotechnology and Microbiology, it would not have been completed. So your participation is a major one for our seminar, and we want to encourage you by giving this poster opportunity. So we'll be getting feedback from you. Thank you, and wish you all a very happy and prosperous new year. Thank you.
Thank you, Arun Muthukumaran. So now we can come to the end of today's session. We'll meet tomorrow at 10 a.m. For the second session of the seminar. Thank you.